Thank you very much, Gordon. Good morning, everybody. It's really lovely to be back here in Split and to meet lots of old friends, and I'm sure meet lots of new ones. So when Gordon asked me to do this talk, he asked me to talk about what I've been doing in the last 10 years. So it might take quite a time to tell you all. So what I thought I'd do is tell you a kind of a bit of a story, and tell you it's a sort of realisation of a dream in a way. You know, one of those ideas that you have at 2 o'clock in the morning and you think it will never happen, and you go back to sleep and forget about it. And then many years later you realise that actually your dream in some measure has come true. So you can judge how well my dream came true. So this is just an introductory page. These are the institutes that I'm fortunate enough to be connected with. And these are some of the molecules that I'm going to talk about. I'm going to talk about exploring pathways that control glycosylation in cancer in multi-omics systems using automated glycoanalysis platforms and AI. So a long time ago, I was working on, began to work on cancer. And I had this question that I've always had about science and molecules and life in general, I suppose. Where does this molecule come from and where is it going? And at the time, this is probably 30 years ago, that was a slightly strange question because science was much more compartmentalised than it is now. So this sort of idea just stayed in the back of my mind. But what I was thinking was that could we link glycosylation with the process of metastasis in some real way? So I started to think about tumours and how they grow and how metastasis begins. So first of all, you have a primary tumour, and the cells from here need to detach in order to move through the extracellular matrix and then intravasate into the bloodstream or the lymphatic system. They then are transported in the bloodstream and they're protected from destruction by the immune system until they can attach and extravasate into another tissue. They then proliferate and they undergo angiogenesis and build their own blood vessels and eventually become metastatic tumours. So my question was, can we trace glycoproteins through this whole process and understand the mechanisms that underlie them at all levels, all the way from the genome down through the transcriptome, through the proteome, down to the glycane? So we started off thinking about detachment. So overexpression of MUC1, and particularly when it has increased O-glycosylation sites, is really important in breast cancer because the ubiquitous cell surface overexpression of MUC1 it increases because the O-glycan sites increase, and also you get increased silation and polylactosamine structures. So these MUC1 structures become very long and very rigid. And because they're expressed all over the cell surface and not just, and in these gaps between, in the gaps between these different cells, they can actually elbow themselves apart so the cells can break the integrin connections and they can become detached. So point number 1, intravasation. So on galectin-3 is a very important lectin. It binds to, one of the things it does is bind to MMP9, which is gelatinase B, which is part of the matrix metalloproteinase cascade. And in cancer, MMP9 cleaves through the extracellular matrix in order to allow intravasation. So how is that affected by glycosylation? So this is MMP9. So the yellow structures are the N-link sugars, the orange structures are the O-link sugars. And these are the glycan structures that we found attached to healthy MMP9 or gelatinase B, or attached to neutrophils, long polylactosamine structures, which are very good ligands for galectin. Galectin binds to these structures and it also clusters. So the idea is the galectin, which is also bound to laminin, can hold this aggressive protease in the extracellular matrix where it's needed. It's not allowed to just go wherever it chooses and cleave up healthy extracellular matrix. But if you look at THP1 cells, you can see that there's much more yellow on this large end-link sugars. There are less O-links, and the O-links that they are very short. So they're not good ligands for galectin-3, so fewer galectin molecules bind to the cancer type MMP9. They're not bound into the laminin, and they're free to cleave apart with the tumour cells to intravasate into the bloodstream. So that's number 2. Number 3, the next thing is these tumour cells have to get transported through the bloodstream, and how do they avoid the immune system? Well, mucins with modified O-glycans on tumour cells display cellulose X. They then associate with platelets and leukocytes via P-selectin. So they cloak themselves with platelets, and it protects the tumour cells from the immune recognition process. So this is a very powerful way of them hiding, so until they reach somewhere that's comfortable for them to land on and intravasate. So when these tumour cells go through the system with the cellulose X and cellulose A ligands, they're looking for P and E selectin, which are in many places, but on epithelial cells, and particularly in lung and in bone, which are the principal places that these cancer cells attack. So here is a cancer tumour cell rolling along with its selectins on the cell surface. And it sees these P-selectin, L-selectin molecules, and the rolling cells slow down. Eventually they stop, and then there's a signalling pathway where integrin is activated. And so the cells are then flattened. They undergo firm adhesion, and the integrin holds them firmly onto the cell surface of the new tissue that they're trying to invade. They then extravasate through the endothelial cell there. They form a secondary tumour, they proliferate, and then they undergo angiogenesis and metastasis. So this is another key process in the transmission of cancer that involves glycosylation. Now, one day, Marion Boland walked into our lab in Dublin, and she was in between jobs, and she just wanted something while she was looking for something else. So she joined us. She didn't know anything about sugars, but what she knew about was transcription, about which I knew practically nothing. So what she was able to show was if she took cytokines, IL-beta and IL-6, and particularly if she took MCF7, which are breast cancer cells, if she took the supernatant, she could stimulate the NF-kappa-beta pathway because it works through the AXL receptor tyrosine kinase to take P65 and P50 and make them into a transcription factor complex, which can then promote the gene expression. And what is the gene expression that it promotes? It's the expression of glycosyl transferases, and it uprates the production of glycosyl transferases. In particular, fucosyl transferase cell, ST36 galactosyl, and GNT5. All of these are needed to make these large tetraantenary silate glycans that we know only too well are associated with cancer progression. So this was really a great gift because this was a plug in this train of events that certainly I had no expertise in. So then we began to think, well, you know, perhaps there is something in this, but we don't have the technology to explore everything. So the question was, can we build technologies to link complex disease phenotypes or quantitative traits like tumour size, disease progression, things like proteomics, and we're talking about the quantity of protein produced, the location. So, for example, the glycosyl transferases, are they upregulated? The polypeptide sequences are also obviously important. And then things like transcriptomics, which involved the NF-kappa beta pathway, but also we showed that hypoxia, we know that inflammation in drugs also changed the transcription of the genes. And then genomics with its association with epigenetics and with this drug, which was used as a drug, 5-azole, a demethylation drug. And then we shouldn't forget the lipidomics and the metabolomics, which are of course also involved in tumour development. So this is my mind map of could we link all these things together by looking at glycosylation. So we thought that we had an opportunity, but we thought now we had enough suspicion that we could do this, to work together with some people who are really expert on prostate cancer and to look for disease indicators. So this is a profile showing the glycosylation changes between PSA from prostate cancer and PSA from control seminal fluid. So you can see that there's a massive decrease in the amount of alpha-1 6-linked core fucose in the PSA from prostate cancer patients. And so this is a very clear marker. So we then went further with this group of people who we decided to look at combining markers from genomics, transcriptomics, the proteome and the glycome together. So we had 4 markers that were associated with genome methylation. We had 4 markers from the transcriptome from breast tissue, and we had 27 plasma proteome markers and 13 plasma glycome markers. And we then combined them all together. So you can see here the ROC curves showing 48 multi-omics markers combined. And so the top, this top profile is showing that the combined markers have got pretty good ROC values for indolent, significant and aggressive types of prostate cancer. And we then compared it with the current clinical markers, which are definitely not as good. So this was quite important to me because I think when you have a disease, there can be all sorts of levels where you actually hit the disease pathway. It might be something wrong with the genome, it might be the transcriptome, it might be a folding problem with the proteins, it might be something wrong with the glycosylation step. Not every patient will enter the disease pathway at the same place. So this rather interested me because I thought this was aligned with something that I thought. So now we have to really seriously think about building technology platforms and also to build glycoinformatics capability because we needed to be able to do it automatically. So in Singapore, we built 4, and in NIBUC, we built 4 main programs. So the database GlycoStore, which I'll show you in a minute, a program called MagMap, which was basically to be able to bring together lots of different techniques and make them into a program where you could get a better indicator of your assignment of glycans. And then the glycoanalyzer, which was built with the New England Biolabs to interpret exoglycosidase array digestions. And then glycopeptide graph mass spec, which we used for doing glycopeptide analysis. So all of this is hosted by Glyspace Alliance in Japan. So this is the program that you probably know best. This is the automated platform for sample preparation, separation and analysis of glycoproteins. So it's basically a release platform and that works on 96 well plates. And then there's an LC part where we take glucose unit values from the elution positions on the LC, and we then run the data into a mass spectrometer, so every LC peak, which has already preliminary identification, is firmed up with the mass spec data. And we then realised that we started to use CCS, ion mobility, and we realised this was another powerful technique. So we now have 3 different techniques that we were combining. And of course you can go on, there's plenty of people here who use computer electrophoresis, Ondresh and Erdmann and various other people. And that also provides a quantifiable measurement of the property of a glycan. So that's all you need, a quantifiable measurement of some property of a glycan. And you can put it into this program, which is a multi-attribute glycan map. And it combines and scores the data, so it increases your confidence in the assignments. So this is very useful. So the next piece of software, I'll just glide over very briefly, but we do enzyme array digestions. They're glycosylation, which are very specific for specific terminal monosaccharides and linkages. So if you take a mixture of sugars that you've released and you treat them with these different arrays of enzymes, you can reduce the size of these peaks each time. If you use a fucosidase, you remove core fucose, and the peak moves down through this set of peaks. So by knowing the enzyme specificity and the shift of the peaks, you can work out the glycan structures, and it's very useful because it gives you linkage analysis without much more problem. So that's the third one. The fourth one is the identification of glycopeptides. Now, one of the molecules that we were interested in was the AXL receptor tyrosine kinase because of its association with the NF-kappa beta pathway. And it's very powerful. It regulates proliferation, differentiation, cancer growth, and migration. So we decided if we were going to look at glycopeptide analysis, this was a good one to start with. It's got 6 N-glycosylation sites. And some of the problem that we found with glycopeptide identification is that there's a lack of quality MSMS data. So it's, you get missed peaks, and you can see in this data here that there are lots of peaks. The red is the original data, and then the conventional database will interpret these blue spots. So you can see there's lots of streaks which are pretty obviously glycans, but they're not identified. And to identify hundreds of spots is a huge amount of work because they all require an SMS. And also you need a comprehensive database. If you're basing your findings on a database, then you have to be very careful you don't miss structures that are not in your database. So when we looked at the commercial software, they identified only 30% of the tumour mark of the glycans in these 6 N-glycosylation sites. So we wondered, could we work out a way of getting more from, getting more from the data. So we use GraphMS, which you probably know from other contexts. But the idea is that from this mass spec graph, we take one spot and we completely analyse it. So it's glycopeptide, we know what it is. We then ask the spot to link up to every other spot on the map, and we get these jumps. The data then will we then ask it just to pull out those spots which are linked to another spot by the mass of a glycan. So this one will link to this one because the difference in mass is 162, which is a hexose. And this one is a hexose, this one is a hexanac. So it selects from all the possible connections, the ones that are related to glycans, and then it draws these graphs. We know the structure of the peptide, and each peptide comes out with these different glycans associated with it. So it's very fast, it's very rapid. And so we then ran exoglycosylase digestions on everything to demonstrate that the peaks that we were identifying were really the glycan peaks that the software was telling us. So when we did this, we used predicted glycopeptide compositions. We expanded our glycan database. We expanded the inclusion list of mass spec predictions, and then it outperforms ordinary database searches, and we use it to find novel glycans. So you can see here that just if you use permethylated N-glycan, at the time 164 sugars were identified, where the GraphMS had 536. So there's a massive difference. You actually know what you're looking for and you go in just to search for that thing. and this is instrument platform agnostic. If you go to Matthew Chu, you can, it's patented, but you can use the patent for free if you're academic. Okay, so now we've got the tools. So the final part was, can we link up multi-omics with a specific cancer, with breast cancer? So we took 62 breast cancer patients and 107 age-matched controls, and we linked levels of tri- and tetra-catenary glycans, which displays sialose X, to breast cancer. So all of these peaks here were increased in breast cancer. And we then linked the increase in this sugar A3F1G1F down here. We linked that to breast cancer progression. So you can see over the course of the disease, this sugar marker is elevated in almost every case. So that's interesting. The serum glycome, again, we linked it to survival. So we looked at GP1, that's one peak down here on the human serum glycome. We tracked that through the months when people had the disease. And we saw that patients who had an increase in GP1 structures had a better survival than those who didn't. So this is circumstantial. We don't know why, but we do know that we can link the glycose sugars to these incidents in breast cancer. But then there's another piece of serendipity. We had a collaboration with Agilent, and Agilent, as you know, really expert in miRNA, mRNA, and transcript levels. So we managed to work with them. We analysed every sugar peak in the serum glycome, and you can see here the sugars that we found in these significant peaks. And then we related them to which peaks were related to survival, which peaks were related to survival of people who were ER positive, ER negative, and those who had basal type cancer. We also looked at tumour size and CTC, so all the red blocks are where these sugars are in significantly higher abundance with an increase in trait, and the green ones are where there's a significantly lower abundance with an increase in trait. And we also looked at glycoproteins associated with mRNA, so these arrows all point to glycans that are involved in something to do with mRNA transcript levels. And these arrows upwards are glycoproteins that Agilent associated with miRNA targets. So this looked amazingly complicated, but Agilent also were able to do enrichment analysis and to work out the pathways that were being activated by this miRNA, mRNA, and through these different groups of sugars. So here, these are the peaks from the serum glycome, so peak 2, 11, 32, and so on. And here are the sugars that are associated with each of these peaks. And what they found was that GP2 is associated with DNA replication and mismatch repair. So they're both involved in cancer, right? And GP11 is to do with fatty acids. This 32 is to do with triglyceride synthesis, and you can read it as well as I can. This is to do with focal adhesion, and then the ones down here are to do with signaling pathways, and another group associated with mismatch repair. And so all of this just looked really amazing, because now we can redraw our original dream of relating systems glycobiology with glycosylation of metastasis. So detachment from the tumour mass because of the increase in the sugars on MUC1, intravasation because of gelatinase B and its loss of lactose immune structures, transport manipulation of the immune system because the tumour cells cover themselves with platelets through sialose X, attachment, extravasation, P-selectin and L-selectin, proliferation, followed by angiogenesis, followed by the development of a metastatic tumour. And then from there, we can then go back from looking at serum glycome, we can say that these peaks are associated with cell cycle control, mismatch repair, protein maturation, and triglyceride synthesis. And we can say that GP36 is associated with focal and cell adhesion, so something to do with the detachment. And GP34 with angiogenesis, GP46 with transport and manipulation of the immune system, and GP46 again with attachment and extravasation. So now, this is like the beginning of, there's so much information here that we really needs to be followed up really, but it's, to me, it's just an indication of how powerful glycomics can be. If you really can get to grips with the analysis and you can understand that you don't have to start with the genome, you can start with the glycome and work backwards just as much as starting with the genome and work forwards. So that's my vision that's partly fulfilled and my vision for the future. So I'd just like I'd just like to leave you with the thought of Newton. It is better to explain all nature is too difficult a task for any one woman or even for any one age. It is much better to do a little with certainty and leave the rest for others that come after to explain all, than to explain